All right. Well, what I'll go ahead and do is again do a little bit of logistics here. Dr. Kerr, while I start doing this, if you don't mind sharing your screen. Just as a reminder, this is not a Peanut Research Foundation board meeting, so we're recording it for a reason. It's a learning information session. So I want to thank you all for joining us in our gene editing town hall here as the Peanut Research Foundation is learning more about the subject and continues its investigation of it as a potential future for us in terms of funding and research. But with that, I'll like to uh, introduce Dr. Parrott. Dr. Wayne Parrott currently serves as the director of the Institute of Plant Breeding, Genetics, and Genomics at the University of Georgia, where he's been named as Distinguished Research Professor. He's received his degree in agronomy from University of Kentucky. Go Cats. I didn't know you were a cat. I'm a University Wildcat. This is why this is going to work. Uh, and an MS and PhD degrees in plant breeding genetics from University of Wisconsin-Madison. Since arriving at Georgia, he's been conducting research on the development, use, and safety of transgenic and edited crop plants. He's published a guide for environmental risk of GMOs, along with 120 journal articles and referred publications in 15 book chapters. He's a fellow of Crop Science Society of America, Society of In Vitro Biology, American Society of Plant Biologists, and American Association for the Advancement of Science. He is actively engaged in training graduate students and postdoctoral fellows and teaches graduate level courses in crop genetics communication technique. He's traveled extensively through Latin America and other countries and advised legislators and regulators in various countries on policies that ensure the safety of genetically modified products or GM products. With that, Dr. Parrott, I turn it over to you. And thank you again for sharing your presentation today.
Well, thank you so much, Chris, for the introduction, and to Reagan for the volunteering me for this talk. But it is a topic that I do spend a lot of time on, so I am rather passionate about. I'm assuming you can see my first slide, and it's not peanuts. I don't work on peanuts, so I don't have a lot of peanut pictures to take. But I took this picture on Friday of last week, so just a few days ago. It is one of our research plots. And what you see here are these trees, and they're all the exact same tree, except some of them have been edited to change the growth forms of the tree. And particularly, we're looking at this morphotype that we call it to produce biomolecular compounds in the leaves. The short ones actually have more leaves in them than the original tall trees do. And the idea is that we can harvest them at the bottom every year, and then they'll just grow back for the next year and another harvest. And I think this photo is just a really good overview and impression of what editing can do. So, sort of the way I'm trying to give a really broad overview of the various aspects of editing. So, because editing is a form of modification, I want to talk a little bit about plant breeding and modification of crop plants, delve into a little bit about the differences between genetic engineering and editing, and then give some background on what happens to DNA during modification, primarily during plant breeding. And that sets the stage for the current regulations. And I want to close off with where things are now as far as the use of edited crops in agriculture. So going back to conventional plant breeding, I have a rice field here, and to make the point that plant breeders with time stack traits. And this is particularly useful data. It's from IR8. It was the first rices, or the first rice variety that touched off the green revolution because it was short, high yielding, and widely adapted. But if you notice, it was susceptible to a lot of diseases and insects. A few years later, by the time that IR42 came out, all these susceptibilities had been converted into resistances. And every one of those conversions required a gene from another source. And right now, I believe they are at IR64. Again, just have a much better package to resist both biotic and abiotic stresses. The thing to notice here about these resistances that some of these were obtained by adding resistance genes, and others of the resistances were obtained by inactivating or getting rid of susceptibility genes. Not that long ago, we used to think of you either have a resistant gene or you don't, but now we recognize that we also have susceptibility genes. Now, since I work on soybean, most of my graphics are soybean. But again, in general, rule of thumb for plant breeding is that some traits are added. For example, in soybean, we have to have the quality traits since it is grown from both meal and oil. We have to have enough pest and disease resistances to make them growable. And we can have value-added traits such as herbicide tolerance. At the same time that we're adding traits, we are, okay, and the genes for these, all of these need genes. They can come from other varieties. They can come from land races. In some cases, they come from other species, and more recently, they can be engineered. They come from transgenes. Now, when we are not adding traits, we are removing undesirable traits. For example, soybean normally has a black seed coat, and when you press the oil out, it stains the oil black, and that doesn't go over well with consumers. So we had to remove the black seed coat. Normally, seed size in soybean is very small. There are actually genes that suppress seed size, so those had to be removed as well. And the genes for these come again from natural mutations that one may find. If one cannot find a natural mutation, say in a land race, then we resort to mutagenesis. And what's in vogue right now to replace mutagenesis is gene editing. So the Food and Agricultural Organization of the United Nations and the International Atomic Energy Agency maintain a database of crop varieties that were produced through mutagenesis, either radiation or chemical. And at last count, there were over 3,000 of these varieties. Here's an example of a mutagenesis facility using cobalt-60. There's actually cobalt-60 in that tower, and the radiation can hit the plants growing around it. And things that have come out of it that you might be familiar with is seedless pink grapefruit, or if you're in the United States, green beans or snap beans. In my grandparents' generation, they were called string beans, but the string is gone, so now you can snap them. Pasta, pretty much any pasta around the world is made with mutated wheat. This is an ad. It appeared in the Chicago Daily Tribune in 1961 when we were a lot more courageous as a society than we are now. And they were selling, Walgreen Drugs was selling seeds that had been radiated at Oak Ridge National Labs. And they were selling, mutated seeds so that people could plant them and see if any novelties could be found among the progeny. And they show a guy here with a really tall petunia plant that came out of one of these. So again, I don't think this would go over well with consumers today. But back in the day, if you adjust for inflation, they were paying about $30 for a packet of mutated seeds. So that brings us up to the present time. I think as a community of plant breeders, we've been really excited about plant editing. An engineering approach can be described as we're bringing the talent in from the outside, whereas the editing approach is we're going to bring out the best in you. In other words, no outside talent is required. The principal tool used for editing is CRISPR-Cas9. Now, there are many different types of Cas. You might hear Cas12, for example. But so far, Cas9 has been the most versatile of all the Cas proteins out there. And Cas9 was actually found in yogurt. If you look at the active cultures, there's S. thermophilus, and that was the source of Cas9. It was some cultures owned by DuPont. They noticed that when they made yogurt, there were some bacterial lines that did not keep the milk from turning sour. They were infected by a virus that would kill the bacteria. And other cell lines were quite resistant to the viruses and would not die off as they made the yogurt. It was trying to figure out what made these cell lines resistant to viruses that they discovered Cas9. But the bottom line is that Cas and CRISPR-Cas has been part of the human diet for a long time. The way that works is we make what we call a guide, but it's actually the CRISPR part of things. And it's got a RNA that matches the DNA that we want to edit and a scaffold. And we have the Cas. The Cas is the one that does all the work, even though CRISPR gets the credit. But it's the CRISPR-associated protein, which is where Cas comes from. And both the guide and the protein snap together. And at this point, they can just latch on to DNA in the nucleus, and they'll just scan all the DNA until there is a match. And once there is a match, it will make a double-stranded cut. Both strands of DNA get cut at that site. And from a plant breeding perspective, it's exactly the same result that we would get with mutagenesis. The big difference is that mutagenesis is totally random. We may have to mutate and then look at 1 million plants to find the mutation we want, whereas with CRISPR-Cas, it is much more targeted. So we do a comparison of what various techniques can do. Here I'm looking at conventional traditional breeding, genetic engineering, and editing. So if we want to get rid of unwanted genes, for an unwanted trait, say a susceptibility gene, conventional can do it just fine. Engineer it will do it really well as well. And editing is the one that really excels at doing this. We want to swap genes, say get rid of an allele from one variety with an allele of another variety. Conventional will do that very well. Say, replace a recessive gene with the dominant version. Engineering will do it. It doesn't get rid of the old copy. It just adds another copy to it. But technology to do this with editing is incipient. It's almost there. We want to insert genes from the same species. Conventional will do that very well. Engineering, this is what engineering really excels at. And it's incipient with editing. And if we want to add genes from another species, it's usually very tough to do with conventional. It's great with engineering, and it's still incipient with editing. So just about all I'll be focusing on for the most part is the deletion of unwanted genes. Just keep in mind that other uses and aspects of editing are rapidly approaching. So for regulatory purposes, particularly in Europe, these different things you can do with editing have been classified into 3 categories. That while these categories are not perfect, they do make a useful framework to think and talk about editing. So the first of these is SDN1. This is simply where the Cas makes a double-stranded cut, and the plant then will come in and repair the cut. But the repair process might leave a scar, and that new scar will either change the way the gene works or more commonly just stop the gene from working at all. So again, this is completely equal to traditional mutagenesis, whereby we're forming new alleles or we're getting rid of a gene's ability to function. The other thing, sort of spin-off from this that we have is called base pair editing, where we might be changing, say, a C to an A, within the DNA code. And again, these are things that also happen in mutagenesis. The next category is SDN2, and it differs from SDN1 in that we're providing a piece of DNA here that can serve as a template. So when the plant repairs the gap that was made, it's going to repair it by copying the DNA in the template. And then, this piece of DNA here is going to end up in that gap. And there's some difference, this template might come from the same species, it might come from another species. But in general, it's equal to backcrossing in a breeding program. We're replacing one allele with another allele. Finally, we have SDN3, where the template is much larger. It might have a whole gene in it as opposed to a part of a gene in it, which again can come from the same species or a different species. But this pretty much is equal to transgenesis, cisgenesis, transgenesis around the world. So again, my talk is going to be mostly focusing on SDN1, which is the technology that's available here and now. So I want to take a step back and talk a little bit about what happens to DNA during genetic modification, during traditional breeding, because that's the baseline that we use for safety evaluations of a given technique. So it's been estimated that over the past century, there were about 1 million new varieties of land races and cultivars that were developed around the world. All of these have a history of safe use when handled properly. We know some of plants are toxic. Some, for example, cassava might be toxic. But as long as you prepare it properly, you don't need to worry about it. Or, potatoes might be toxic as long as if they get green, but if you don't let the potatoes get green, they're safe to eat. The safety record comes despite the fact that very few safety studies have been conducted on any of these plants. And the other thing to keep in mind is that for the most part, it is impossible to change a trait, to change the way any plant is going to look without changing the DNA that's inside. So every change we make on the outside, there is a change in the DNA on the inside. So the question from a safety perspective, then, if we consider the changes that happen in breeding to be safe, then for the question to ask is, how do the changes made to DNA by editing compare to those changes that happen during breeding? And that is something that we can start to address with genome sequencing. And I'm going to begin with mutation breeding. So when we do plants that have mutated, we find deletions from one base pair all the way to 8,000 base pairs. We find frame shift mutations that might change the amino acids. We find premature stop codons, which truncate a protein before it's fully made in the plant. We find inversions in chromosomes, whereby a segment of chromosome just flips orientation. And we can find insertions of 200 base pairs or even larger. And when we look, and we know then, since we've been doing mutation breeding since the 1930s, that these types of mutations have all been safe. None of them have had a food safety or an environmental safety issue related with them. And it turns out that these same changes are like the ones that happen during the editing process. Now, during breeding itself, the advent of sequencing of genomes has really given us unprecedented view into how DNA changes. And there's a lot more changes than I think any one of us ever would have imagined possible. So genetic modification, meaning plant breeding and everything else, we know that modification is done by, and we add removed traits by changing the DNA. And these are the two most common types of changes we see. We see SNPs, whereby one DNA base pair is converted to another DNA base pair. And we see indels, which stands for insertions or deletions. We don't really know if we added base pairs here or if we deleted base pairs there. And then, as I mentioned, we have segments of DNA that can just flip orientation or even move from one chromosome to another. And both mutagenesis and SDN1 mainly create indels and SNPs. SNPs can have a large impact on the phenotype. The photo I showed you at the beginning of the various trees, all those changes in shape were caused by one or two different SNPs. The work that I've been doing for a long time of insect resistance in soybean, the difference between these two soybeans is one SNP in one gene. Modern wheat is a result of two SNPs. Here's an old painting, 1865, of a veteran in a new field. And the thing to notice is how tall the wheat was. Now, I don't think any of you in this call have seen wheat this tall. Wheat these days is rather short. In fact, there was this article in the local paper that I thought was kind of insightful. All of you have heard America the Beautiful with the amber waves of grain. Those amber waves of grain, that was not modern wheat, because modern wheat is too short. So modern wheat sways in the wind, but it doesn't form, it doesn't sway, it just nods a bit. But anyway, SNPs can be very powerful effectors of change. But it turns out there's a lot more to diversity than there's SNPs, and there's a whole new category that I think nobody realized how extensive it was, and it's called structural variation. I'll give you an example of structural variation. Here we have DNA from a segment of chromosome 10 in round tomatoes. It turns out that this part of chromosome 10 got copied and moved onto chromosome 7, where it interrupted a gene that was there. And the result was the long tomatoes. And we think this happened in Spain probably about 400 years ago. Wine, I'm sure some of you are aficionados of red wine and others prefer white wines. Well, in this case, a Cabernet Sauvignon, you add a piece, a retro element into the gene for purple color, and you get your Chardonnay. Or here's corn. Corn is normally white. You have an enzyme called phytoin synthase that makes phytoene, and this accumulates, here's the gene, the phytoin synthase. This accumulates, this is not made, it accumulates in the leaves, but it turns off, it's not made in the kernel, which is why the kernel is white. Again, an insertion into this gene prevents the phytoin synthase from turning off. So it's still it's on in the leaves, but it will not turn off in the seed, allowing the seed to make carotenoids, and hence a change in expression pattern. And these are the same type of changes then that we can make with editing. Soybean seed color, I mentioned this. Soybean has a series of 6 genes. It's actually 2 or 3 genes found in 2 copies, and they're mirror images of each other. If we remove one of the copies, the seed coat turns black. Again, we can do this type of removal with gene editing. Spicy but tasty. The pungency of chili peppers. I don't know if anyone's ever tried a chili pepper. I could have been that kid when I was about 4 years old. But again, here's the gene for pungency, and when the sweet peppers have lost the gene, which again is a type of change we can make with editing. Then there are other types of changes. Some of them are called like copy number variation. Since we're in peach season, that's a really good example to use. Everyone's familiar with clingstone peaches, the ones that flesh clings tight to the pit. Those have melting flesh, and the melting flesh is caused by an endopolygalacturonase enzyme that digests the cell walls. We have freestone peaches. I think the name is self-explanatory. And in freestone peaches, the enzyme has duplicated. And then we have clingstone that does not have melting flesh. The hard peaches are used for canning purposes. And there, the gene has gone altogether. So the number of copies varies from 0 to 2. And then we have presence-absence variation, where genes can be there or are gone entirely. So looking at tomato varieties, I think they looked at 725 tomato varieties, and they had found some that only had about 35,600 genes, and other varieties had 37,400 genes. So in general, they cataloged about 5,000 genes that may or may not be present in any one variety. And these 5,000 genes then are the ones that get mixed and matched by breeders to come up with the different tomato varieties. And wheat is the one that takes the cake. The average wheat variety has about 130,000 genes in it. Of these, there's only about 81,000 that all wheat varieties share, which means that there's about 50,000 genes that any variety may or may not have. In fact, they have cataloged about 140,000 genes from that may or may not be present in the different varieties. And again, that's what we mix and match to create new varieties. So this kind of summarizes what we see. Here's a study in barley. They looked at a barley variety from England or Europe, one from the US and from China. So if you compare the US and the European one, they differ by almost 14 million SNPs, and they differ by over 1 million indels. Comparing the Chinese with the European, again, over 11 million SNPs and over 700,000 indels. And comparing the US with the Chinese, over 13 million SNPs and again, over 1 million indels. And yet they're all barley. So the thing to keep in mind is that if the first few million SNPs and indels did not affect safety, any possible effect from one or more SNPs or indels that we would do with editing is going to be too low to measure. And as I mentioned, this is going to come and influence regulatory policy. So the key points that I want to make so far, that I think I've tried to make so far, are that changes to DNA are the basis for genetic diversity that we depend on in breeding. Changes are very abundant, and there's a lot of diversity. And gene editing recreates changes like these, right? At the present, SDN1, we're using a lot, and in the future, we're going to see a lot more of SDN2. And this is across plants. So what that means, or from a safety perspective, is that the type of changes being made by plant editing have a long safety record in plant breeding. And if there's any risk, it's not going to be from the change to the DNA, it's going to come from a trait. So, any safety assessments need to be trait-based. You might add a new protein, create a new protein. Does it cause allergenicity, for example? So this, I hope, have set the stage for regulations. And in the United States, we have what we call the Coordinated Framework for Biotechnology. The framework has 3 government agencies to it, and historically they have not been nearly as coordinated as they aspire to be. But the first of these is the USDA, and they regulate plant pests. And they've defined, what they did is, because in the early days of genetic engineering, we were using DNA from plant pests. The USDA decided that if you took a little bit of DNA from a plant pest, added it to a plant, the whole plant would become a plant pest, and thus they could regulate it. And they've been backing off that stance in the more recently. FDA has authority over anything that's going to be used as food or feed, and the EPA can regulate pesticides. And I'll get to them one by one. Now, as additional background, any product that contains no transgenes, because we do use transgenes to make the edit, but then we get rid of them. So as long as the edited product contains no transgenes, it's just not a GMO. It still is in Europe, but most of the world it's not. But because it usually has a GMOs, but again, it usually has a GMO step. So making GMOs is still very variety and species dependent, which can be laborious and expensive to do. And the other thing that we need if we're going to come into an editing program is we need to have excellent genomic resources so that we know what genes to be targeting with our CRISPR-Cas. Now, the USDA does not, or none of the US agencies use SDN terminology, but if you assume that no DNA is introduced during editing, in other words, if we're dealing with SDN1, and we can do that in a seed propagated crop like peanut, you simply segregate the CRISPR-Cas9 out and you leave the edit behind. Or the other case is we did introduce DNA, but as long as that DNA does not come from a pathogen, or as long as that DNA was not introduced by a pathogen, meaning Agrobacterium. In other words, we're dealing with any of the SDNs in this case, but the difference depends on the source of DNA. Edited plants are not regulated by the USDA. And one can self-determine, it is legal for you to say, my CRISPR-Cas has been segregated out. There's no other DNA that got introduced. I'm not regulated. Or if you lack the courage of your convictions, you can write the USDA an "Am I regulated" letter where you say, we use CRISPR-Cas and it's been segregated out. There's no other DNA left. And they'll send a letter back confirming that you're not regulated by the USDA. And what I did is I went through the "Am I regulated" letters and started pulling out some of the "Am I regulated" questions that have come through, because that gives an insight into products that are being developed in the US and are somewhere in the production pipeline, everything from non-browning avocados to better tasting peas, easier malting barley, low phytate canola, virus-resistant tomato, non-browning lettuce. Actually, this one's already on the market. Better tasting mustard, canker-resistant citrus has just entered the market. Non-browning potatoes, acrylamide-free potatoes, novel-colored petunias, high oleic oils. We have a jointless tomato for mechanized harvest, altered plant architecture, altered nutrient composition, altered maturation time, altered flowering windows. Thornless blackberry, it's already commercialized in South America, as is the seedless blackberry. I'll show you some photos. This is just a different company looking at altered flower color, non-browning bananas, herbicide-tolerant cottons, and so forth. So we are actually seeing a lot more edited plants coming through the regulatory pipeline than we are seeing genetically engineered plants. Moving on to the FDA, there's two aspects of FDA, one that regulates animals and one that does plants. Now, for farm animals, according to the FDA, they say that the portion of an animal's genome that has been intentionally altered, whether recombinant DNA or modern gene editing technologies, is a drug because it is intended to alter the structure or function of the animal and thus subject to regulation under our provisions for new animal drugs. So basically, the farm sector is just out of luck with editing because getting the FDA to approve an edited animal takes hundreds of millions of dollars because it has to be tested as a pharmaceutical. The story for plants, which is another part of DNA, is much better. In general, all food sold in the US has to be safe and it has to be legal. And if you have any doubt about the two, you just consult with FDA. FDA can regulate traditional breeding of all crops used as food or feed, but historically has only focused on biotech crops. Now, consultation for safety is voluntary, but I put voluntary in quotes because many times, even if FDA does not make you go through it, the market will not touch your product unless it's been through FDA. And when it comes to editing, FDA is strongly encouraging consultation, particularly if any proteins are involved. Finally, that gets us to the EPA, which regulates pesticides. And pesticides are defined by intent. So here I have a genetically engineered potato, the white russet, which is resistant to the Irish potato blight. I have pyrethrin, which is a common insecticide. And I have a Samsung dishwasher that is will kill bacteria when it washes clothes. All three are registered pesticides with the EPA because all of them intended to kill something, whether it was the blight fungus, whether it was the insects, or whether it was bacteria. So again, any plant that's engineered or edited for pest or disease resistance is therefore treated like an insecticide. And the safety testing and the rules to approve these are the same for all three items you see here. Doesn't really distinguish between a disease resistance gene, a traditional insecticide. Now, if the edit results in disease or pest resistance, you have to go through EPA. Otherwise, EPA is not involved. So you're allowed to have one SDN1 edit if you consult with EPA. And if the sequence is identical to what is in the germplasm pool, or if the change is possible by convention breeding, they will exempt you from registering your product as a pesticide. They will allow an SDN to edit, again, with consultation, if the source DNA, what you use as a template, is a cross-compatible species, and if the DNA sequence and expression pattern is identical to what's in the germplasm pool. And if you don't meet these criteria, you have to register as a pesticide. And I think that's one reason we don't see a lot of engineered plants that have been engineered for disease resistance, is simply getting them registered as pesticides can be worth more than the crop itself. Another criterion is that your product must be exportable. And here in green are countries that are favorably inclined towards edits, that these countries recognize the edit as being different from an engineering or a GMO. And the vast majority of them, you have to run your edited product past the government, get them to certify that, yes, indeed, it is only edited, it is not transgenic, and then you can enter the product into the country's market without a problem. Mexico is still very antagonistic. Europe is still very antagonistic. It says here it's changing its regulation. They just voted to change their regulation, but they have set 2 years time to actually change the regulations. And until we know what they change them to, it's going to be very hard to say whether Europe is going to open up to editing or not. Then here's just a book that goes over the different legal frameworks for editing around the world. I'll just say it's a moving target. You know, the field of editing regulations is evolving and changing very, very quickly. So despite all the regulations, again, a key point is, though it may look like there's many exemptions from GMO regulations that are being made for edited products around the world, every single country has safety laws to apply, or that apply to all foods, and they have environmental safety laws. And I belong to the group that think we didn't need any biotech regulations at all, that the safety of biotech food would have been perfectly covered under existing legal frameworks around the world. And again, no edited product is exempt from the overriding safety laws that apply. And that also is true of all breeding. No conventionally bred product is exempt from any of these safety laws, which is why you would not want to market, say, a toxic potato or an almond that has high levels of cyanide in it. Something that affects markets. Edited crops cannot be organic, and that's both domestically in the US and internationally via IFOAM. And according to their criteria of 2016, genome editing is an excluded technique. According to their criteria, it says the genome has to be respected as an indivisible entity. Technical physical insertion, deletions, or rearrangements of genome is refrained from. And again, this would be, particularly the editing would be some of the stuff we're doing with editing. So that runs us contrary to their criteria. And I do find it ironically that nature is the largest changer of genomes. Nature does all these things all the time, but as long as you do it as nature does it, it's okay. But if we do it in the lab, it's not okay for organic. And finally, this is still an evolving area, and that is labeling. The U.S. has bioengineered labeling requirements. They have been in place for about 4 years now. And so edited plants are not labeled if they could also have been created through conventional breeding. And what we don't know at this point in time is where they're drawing a line. If you edit 1 or 2 genes, yeah, probably not. If you're editing 10 genes, yes, it could have been done by conventional breeding, but it would have taken, mutations of tens and tens of millions of plants. So it's not likely anyone would have done it. So anyway, we're still waiting to see where they draw the line. So finally, what's out there? The Japanese were the first to beat us to market with CRISPR-Cas. They have a tomato that's high in gamma aminobutyric acid, which they claim lowers blood pressure, can relieve stress, improve sleep quality, and protect skin health. And you can buy it both as fresh fruit and as canned juice in the supermarket. This product is not legal in the United States because it's making health claims. Lower blood pressure, for example. And in the US, if you make health claims, you have to have data to back it up. Now, the first edited product, there's other ways to edit besides CRISPR-Cas, was actually the Calino premium cooking oil that's high in oleic acid. Cover cress, it's a new crop that's being sold for cover crops that can you can harvest both the oil and the protein meal. One of the limitations to adopting cover crops in the US has been they cost money to plant and then they produce no income at the end of winter. So here, this is a cover crop you can plant, and then when it's summer planting time again, you harvest your crop and you have marketable oil and protein. And they did a lot of editing for things like knot shatter, oil quality, architecture, and whatnot. Pairwise, they came out with I guess Dr. Baker might talk more about this. They removed the bitterness out of mustard greens. And now they have their seedless blackberries, which I understand are in production and is sold supermarkets in Colombia. But they did more than seedless nuts. They made them thornless, and they changed the size of the brambles so that now you can grow them in beds. And so they're much easier to plant and to harvest. So again, it should be production should be a lot more cost effective. So again, it's another idea of just changing the whole architecture of the plant for more modern production methods. Green Venus is selling a lettuce that doesn't brown, so it has better shelf life and better eating quality. And they're also making fast-growing lettuce to use in controlled environment agricultural systems. I mentioned the University of Florida has edited a canker-resistant orange. This is the first disease-resistant edited product, and it is the first product approved by EPA. And Simplot has, a remontant strawberry coming soon In other words, it will bloom throughout the entire and set fruit in the entire growth season. So you can see a lot of things are on the way. So anyway, that's my overview of editing, and I hope it's the type of information you were wanting. So with that, thanks for your attention.
Thank you, Dr. Parrott. Very insightful. I would like to invite Dr. Baker and yourself to stick around, come off camera, Dr. Baker, to just do a little bit of a scientific panel. So really what we're doing, we just learned about gene editing and its aspects, and we're going to kind of shift the conversation. What could this mean for peanuts? Where the highest value opportunities may be, and where it takes research to impact, and how the Peanut Research Foundation should think about investment partnerships and responsible adoption of this. But before we start that, Dr. Baker, I'd like for you to just give yourself a quick introduction, please.
Sure, Chris. And I enjoyed Dr. Parrott's talk. I thought it was super informative. I grew up on a farm in Washington State, and then I had to work a lot when none of our tractors had cabs. So education was kind of the way out for me. And I have a PhD, and then I got an MBA. I ran genetic. I've done some investing early in my career, so I was on the financial side, and then I switched back over to science. I ran genetics at Simplot, so I started that strawberry program that Dr. Parrott mentioned, and then I did that for 8 years. I worked in potato. I launched all the potato biotech products. Built the team, or I didn't. I built the teams that did. And then I was a co-founder of Pairwise, which is one of the larger CRISPR gene editing companies in the world now. And my partner took over a couple years ago. I had to take a break as I stepped back into it. I'm on several boards on a number of these crops, and I'm fairly familiar with most of the things on the market. Most of the CEOs of these companies are friends of mine. So happy to talk about peanuts today and share some things I've learned over the years from commercialization.
Thank you, Dr. Baker. So Dr. Parrott, we'll start with you. Looking specifically at peanuts, where do you see the highest value opportunities for gene editing?
So unfortunately, I'm not as familiar with peanut as I am with other crops, but to the extent you have susceptibility genes, I think you might be able to change maturity dates, maturity groups for either shorter or more extended flowering seasons. Oil quality is always an issue. I know recently ARPA has been talking about the peanut as a biodiesel crop. That would be another change. So just that's a few to start with.
Dr. Baker?
Yeah, I mean, this question on, low-hanging fruit or highest value really depends on where you sit. So if you're a startup company, you want to get you want to get someone to pay you early for it. If you're a farmer, you're generally interested in yield or yield protection. How do I lower my cost or get more per acre? And then I think if, I barely understand peanuts, I'd say, but if you're a sheller, you don't want to be segregating all these different crops. And if you're an end buyer, it's got to fit the sensory, everything you buy. And so a number of these products have been sort of niche markets, so stay out of the mainstream commodity stream so they don't affect everybody. But if you wanted to say, where could you get a quadruple win from the trait company all the way to the end buyer, it's going to be something that doesn't change the peanut, but increases the yield probably or lowers the cost. I think roots are probably, I'm not saying there's traits there, but roots are for drought or water efficiency is probably one of the places to think about. I think what Dr. Parrott said is also correct. And then honestly, it's not just EPA. Disease is hard, but there are some pretty consistent disease challenges. And maybe the technology is not ready right now exactly for every disease, but it's probably coming over the next 10 years to work on that. So I'd like to see industry alignment, and that's generally going to be around a lower cost product or higher yield product.
Thank you for that. So just kind of pivoting to lessons learned from moving gene-edited products from laboratory to the marketplace. Anything that we should understand as a peanut industry? Dr. Baker, we'll start with you.
Sorry, me, Chris?
Yeah, you,
Yeah.
Dr. Baker. Yes,
Okay.
please.
Yeah, sorry, my Wi-Fi went out. So, well, let's just talk about mistakes people make, and then maybe talk about, some of the things that have worked well. Typically, you see the universities stumble over the intellectual property piece. Generally, no one enforces patents while you're doing research in a university, but when you try to commercialize, you could be 5 or 10 years down the road, and then all of a sudden you need licenses and things like that. And that's been a tricky part. I've watched how UC Davis and University of Florida have tried to sort out the IP landscape. And I don't want to, the CRISPR Cas things, you've got to navigate it. I'm not saying it's not impossible. You just have to think about that before you get too far down the road. And then the place that the small companies usually stumbles on the germplasm selection. So none of this stuff is good enough to make up for bad germplasm or bad varieties. So picking the right variety to start with is also super important. So those are some of the mistakes that have been made. But I think that one of the lessons learned is, and I've got a friend that runs a company that works on soy commercially, and they're fairly successful, but for every target they choose, like let's just say they wanted to choose more roots or more root density, even they would choose 3 or 4 genes per target, and they would choose 3 or 4 edits per gene. And so it's a lot more comprehensive than just let's go edit one gene once and see what happens. And so I think having a low enough cost to be comprehensive in your search, you're better off doing a few things well than trying to survey broadly. And I think that's been, there's probably been a couple $1 billion that have gone into this now, and that's where I've seen success.
Dr. Parrott, how about yourself? Laboratory to marketplace. What should we understand today as a peanut industry as we try to maybe move forward with this technology?
I definitely agree and support with the comment on intellectual property. Both understanding what's already there and the ability to create new property, particularly the university IP offices are simply way underfunded, but also be thinking about what your edits are going to be doing, what type of regulatory environment they're going to face. You may be looking at insurmountable price barriers there. And then the other thing that we do ask is that I think all scientists, particularly if you're doing editing or engineering, need to pay attention to the concept of stewardship, which, among other things, means being respectful of a community's values and fears. Be transparent in what you do, and just don't think, just because the USDA doesn't regulate this, it's okay to mix it in with other peanuts. So we have a lot of education to do in that aspect.
Thank you for that. I kind of sticking somewhat to that theme. Dr. Parrott, we'll start with you. If the Peanut Research Foundation could only fund one area related to gene editing over the next 3 years, where would you invest those dollars and why?
As I mentioned before, we still use a genetic engineering platform to do editing. And I haven't been keeping up with peanut, but I gather that it's still not routine or easy. So we really need to make a push to make peanut a lot more engineerable than it is now.
Can you say more about that? What does engineerable mean to you, I guess?
So generally speaking, engineering has 2 aspects. All biotech is done at a single cell level. So number one, that cell has to be able to accept the incoming DNA. And once it's accepted the incoming DNA, you have to get an entire plant back from it. An entire normal fertile plant back from it. And there are very strong genotype effects from it, and efficiencies tend to be low. So, over the past decade, I know we've done a lot of lobbying in Washington. And they have been funding a lot more just basic biology research to improve genetic engineering. There have been a lot of breakthroughs, the use of regeneration genes or morphogenic genes, there's use of viral vectors and everything else, but these have not been applied to peanut.
Great, thank you for that. Dr. Baker, about yourself, next 3 years, Peanut Research Foundation, what should they fund and why?
Well, I think that it's tricky. These dollars are so precious. And so I'd hate to see precious dollars trying to duplicate things that have already been solved. And so that, I mean, I don't know how many academic labs do peanut gene editing, maybe 2 or 3. There's at least 4 companies working on it. And so I'd hate to see you fund something that one company solved, but the academics are trying to keep up, or vice versa. The academics have solved and the companies want. So and since it's not completely transparent where everyone is, I think regeneration's been solved in some places personally. But I'd love to see more of an X Prize format, Chris, where you guys figure out what you want, whether it's 10% more yield or 10% less chemical, however you want to make it. And then whoever has a product wins it, rather than trying to pick a winner and 10 losers, because everyone that applies to grants is trying to get your money to fit their existing research. I'd love to see a voice on what the industry really wants, and then let the people compete for it. Might be an interesting way to think about it.
This is our first step in that journey to try to create that singular voice or what we want. So, Dr. Baker, we'll stick with you. What's the biggest barriers for gene editing peanut from a research plot to commercial production and ultimately into food? So what's the biggest barrier you see there?
Well, I mean, I've seen companies that I've been involved with, I don't know, they have 80 or 90 traits in the field, probably combined. And so there's a lot of them, and that's after they worked in a greenhouse. So there's a lot of 3 to 5% yield traits. And so it's really tricky. You have to do a lot of trials to sort out whether a 3% yield trait's real. There aren't very many 10% yield traits. Maybe I've seen one in my entire career, 1 or 2. So low real but low yield improvements are actually very difficult to you have to keep trialing and get bigger and create the seed to actually test. So I think like the primary barrier is it's from research plot to commercialization is if you have the germplasm right and you have the intellectual property right, is having a trait that's you can easily identify and easily add market value. The market value is missing more often than the other parts of the regulatory or the science, if we're going to be honest about it right now.
Parrot, how about yourself? You mentioned a lot of regulatory concerns and other things out there. What do you see being the greatest barrier?
Public acceptance,
Hmm.
which again, what we're seeing is that if you have a trait that has something to offer the end consumer, the one that does the shopping in the supermarket, if it offers them a direct benefit, say like the pink pineapple does or the purple tomato does, they have no qualms. They don't care if it's engineered or edited. But if the benefit to them is not obvious, you do see pushback. So, starting with a trait that I think has consumer appeal, if you can identify one, I think that's where I would start.
But related to that, I guess, a trait or yield or picking a trait, what should peanuts do early to improve their odds of that real world adoption? Is it a consumer trait or is it yield or, where do you see that? We'll start with you, Dr. Parrott, on that one.
I would go with the consumer trait.
All right, Dr. Baker.
Yeah, maybe, give me, real quick on the acceptance piece.
Hmm.
I agree with everything Dr. Parrott said, but I maybe add three more dimensions to it that weren't mentioned. First is in small amounts, these gene-edited products are difficult to detect. They're not at all like GMOs. When they get mixed in, it's pretty difficult for the most part. Depends on the product. The second is we haven't really talked about how AI is changing this,
Hmm.
which is an interesting conversation, probably a separate whole meeting would be really interesting to talk about that. But the last point I wanted to make is that, when we a lot of the times we're looking back at GMOs because what happened with GMOs, we're imposing gene editing on it. But the reality is the acceptance piece around gene editing is being driven in the human health area. And the risks we would suffer in agriculture are much, much stronger in the human health area probably than they are from the agricultural side. And so, for instance, like we have gene editing cures for sickle cell anemia and deafness and many other genetic diseases coming, low LDL cholesterol. These are all in clinical trials. They're moving fast and they're genetic cures. So yeah, you have this halo with curing diseases, which is really nice. We're going to cure blindness. But the flip side is that every once in a while, there's a story about the Chinese engineering babies. Right? And so and being much more, we never saw that with GMOs. You might have saw an animal, but engineered humans is something that's a risk factor out there with the CRISPR technology. And so I think we just need to watch that human health lens. And so um, I have not seen, I think people like the talking points of these consumer traits. Europe's chosen to go after climate change. So when you see this European perspective about how did they get behind this law change, it's about the changing climate. I think it's a little more controversial here. But but industry can't get the economics aligned usually around these consumer products. I mean, they're too various. So generally the yield has been better for seeing success that benefits everybody. So anyway, that's my perspective.
Thank you for that. We've got about 5 minutes left for us to stay on time. So I'll start with Dr. Parrott. It's 2036. We're doing this again. We're sitting around and asking you, about peanuts. What would success look like for gene editing in peanuts 10 years from now in your mind?
Oh, having edited varieties on the market.
On the mark. Okay. Dr. Baker?
Well, if this follows other forms of technology, Latin America is going to adapt quicker, faster, and deeper than the U.S. So, I mean, and in soybean research, if you look at soy trait research, you just take it straight to the field in Argentina, Brazil now. It's cheaper there, it's faster in the market, it's more accepted. So, it would be nice if U.S. has kept up with Latin America, I'll be honest, on peanuts or any of these crops. And then secondly, I wouldn't be surprised if 50 or 60% of the peanuts in the US had a trait. I wouldn't be surprised if there were 40 or 50 in development. I think they're going to be a part of every breeding program. So that's the thing we didn't get into is right now you're talking about adding a benefit to a variety. This is going to get integrated with it already is in some crops, but it's going to be an integrated part of breeding programs. We just have to get the cost down. And I guess that's if I could wish anything for the peanut industry, independent of my own interactions, is how do we get this cheap enough so everyone can use it?
Well, with that, I want to thank you both for your time, and Dr. Parrott for the presentation, Dr. Baker for joining us on this panel. So I think what I've taken away from this is that, it's not just a technology that we're looking at. We're looking at a strategic initiative here for the Peanut Research Foundation. So hopefully with y'all's insights and what we just learned, we can ask the right questions on value creation, what is the right peanut, what is the right, all the right traits and all these things moving forward. So with that, I thank you and appreciate y'all's time. With that, that does take us to a 10-minute bio break. We're running about 2 minutes ahead of schedule, so we'll still go ahead and do that. And we'll come back with our industry panel. And I'm sure our industry panel folks is taking some of the stuff they just heard and revisiting their answers and their questions. So thank you for that. And we'll be back in about 10 minutes, folks. Well, folks, welcome back. like to get started again with our industry panel side, and I'd like to say thank you to these folks that you see on screen participating. But first, again, thank you to our expert panels for, kind of giving us a strong foundation on, what is gene editing, the science, and where it's headed. For this next discussion, we're going to kind of shift from the science to more of itself, the science itself to more of an industry perspective. So we do have, anyone works on peanuts. Those who don't work on peanuts may not know who these folks are, but we have everyone throughout the value chain represented here, from Dan Ward in the farming side of things, Reagan Wiggins on shelling. We got John Moore, kind of sits in the middle between farmers, shellers, and the customers and things of that nature. And then Josh Rahn, a manufacturer with the Hershey Company, to kind of share. So really what we're trying to dive into is this, as this technology becomes a practical tool, where does it create value for us as industry? What problems could it help us solve? What concerns do we need to address? And what would the industry need to feel confident considering adoption? It is setting some ground rules, not intended to be a technical debate. It's a practical conversation about the value of it, the adoption and alignment as a peanut industry. So, as I ask this first questions, if you don't mind reintroducing yourselves and just giving a little bit of background of yourself. And we'll start with Dan Ward on this. From your segment or point of view of the peanut industry, what is the biggest challenge or opportunity where you find peanut genetics could make a meaningful difference? Dan.
Hello, everyone. I did not get a chance to introduce myself. I got a little bit late to the party. And so I'm Dan Ward. I'm from southeastern North Carolina. We grow peanuts and served on Peanut Foundation, serve on sustainability, serve on the Peanut Growers Association, do a little bit of everything. But to answer your question, Chris, I think the number one thing we need to keep in mind from a farmer's perspective is we want to use this technology to increase peanut profitability. And we can either do that we can all say yield. But I think the profitability, we can work towards a number of diseases. We can work towards tomato spotted wilt. We can work towards leaf spot. We can work towards sclerotinia, white mold. We can work towards, what was the other one I wrote down? Root knot nematodes. We can work towards root knot nematodes. So any of those we are working on. And so I think this is a natural continuation of what we're working on. So back to you, Chris.
Reagan, same question. Please introduce yourself.
Hey guys, I'm Reagan Wiggins. I'm the research manager from Premium Peanut. We are a sheller cooperative, but I'll try to keep most of my answers pretty consistent for all shellers. So to kind of talk about this question a little bit, some of the things that Dan mentioned, any of those production improvements that help a grower can funnel benefits to us as shellers, things like disease resistance, that reduce plant damage and improve yields, gives us as shellers more high quality product to be able to shell and to sort. And that ability to be able to reduce input costs for growers from spray applications by planting resistant varieties can make a really big difference, especially considering the current economic climate that growers are facing, and really everybody across the industry. Another big one is the proliferation of aflatoxin. So anything that can reduce our risk of aflatoxin, shellers really kind of shoulder most of the burden from aflatoxin contamination. So things like drought tolerance or things like some grade factors can also help reduce the risk of aflatoxin contamination, like LSK reduction. So I think a lot of those post-harvest traits are important to consider as well.
John Moore, glasses are off. You're ready to go.
Oh yeah, don't have to read now. I think all of those are great ideas. The biggest problem we have in the peanut industry right now is a drop in consumption, soft consumption. GLP-1s have really seemed to kind of take the wind out of our sails, and using gene editing to improve the peanut for the consumer as well as, working on disease resistance and yield increases. I think if you can increase fiber or increase protein, make the flavor more robust. Anything that you can do to make it more appealing to the consumer and drive consumption a little bit. And also with that, make the consumer more, kind of put the onus on them to accept this. Like, we're trying to make a better peanut for you. So, please accept this science and get on board with us. And, hopefully that's the way we get over the hurdle of everybody looking at it as a Franken peanut, something built in the lab. So, I think, like I said, higher fiber, higher oil, higher protein, more robust flavor. Those are all things that I think, and talking to people really on the buy side in this case, want to see out of gene editing.
Thank you, John. Josh, quick intro of yourself and your POV, the biggest challenge or opportunity for peanut genetics.
Okay, thanks, Chris. My name's Josh Ron. I'm a staff scientist for the Hershey Company. Just getting my feet wet back in peanuts. Started about 6 weeks ago. I'm a former Martian, so I worked on the peanut science team over at Mars for quite a bit. So to answer your question, some of the biggest challenges, I think, from the manufacturer side, and obviously we're going to have more discussions on, alignment across manufacturing to make sure we all are on the same page. But a lot of the problems haven't changed over the years, and some of them have only gotten worse. So, for example, climate change, so drought tolerant peanuts, heat tolerance. We'd love to see some areas in reducing the aflatoxin. Wouldn't it be great if, aflatoxin segregation went away? Disease resistance, which was, already described. Consistent quality, so, things around purity. And then overall, having a resilient supply chain. So, having a supply chain where you can poke some holes in it here and there, and you don't spend a lot of time repairing those holes. You're able to smoothly transition from the farmers all the way through the shellers and through into manufacturing and the consumers. Thank you.
Dan, going back to you kind of mentioned a few traits already, but if gene editing could help address 1 or 2 production challenges, you listed like 4, 5, or 6. What traits would matter most to the growers and why?
I think the root knot nematode overall would be one of the high points for the growers. You would save on your insecticide. It's difficult to control, but also something that would matter to all or most growers. Most growers would be your leaf spot or your white mold resistance. I think those were 2 of the ones we targeted through the original genomics. When we started the genomics, we used the figure that if we could come up with complete leaf spot resistance through our genomics packages, that we would be able to save the growers. At that time, it was $52 million a year through the money that we spend on fungicides in the field for leaf spot. So if we could just say, let's just take leaf spot, it would be one of our number ones. We are already working on it, and we've got a lot better resistance than we used to have. But if you had complete leaf spot resistance or pretty much complete control, it would really be a big benefit to the grower community.
Thank you, Dan. Reagan, you mentioned quite a few farmer traits yourself earlier, but what, in terms of a sheller perspective, would be the most value? Consistency, disease reduction, grade size, handling quality, or something else in your mind?
Yeah, so I'm going to speak specifically from a sheller's perspective. I think one of the areas that we don't really talk about too much is post-harvest qualities, or things that impact quality post-harvest. If you're not a sheller or somebody who manages a buying point, it's really hard to conceptualize what a farmer stock peanut warehouse looks like. But there's 15,000 pounds of unprocessed peanut farmer stock that are stored in warehouses for up to a year before they're brought to shelling plants. And so one of the biggest things that we have to deal with is preserving that quality from the farmer's field before that processing step at the shelling facility. And there's lots of things that affect that quality. So a lot of those grade factors are things that we look at. So anything that can reduce LSK percentage or improve the percent of mature kernels that we get into those storage warehouses, or that can prevent those peanuts from incurring any additional quality loss in storage, whether that's from insects or aflatoxin proliferation. I think a lot of those are also very important characteristics to look at.
But John, you didn't introduce yourself, so you're going to get a chance to introduce yourself with this question. But also, you mentioned, like the marketability / consumption of peanuts, trying to rise those. But what would buyers need to understand before they were comfortable with peanuts developed through gene editing?
All right, sorry I didn't introduce myself. John Moore, Albany, Georgia, peanut broker, like pina coladas, getting lost in the rain, not into health food, but I'm into champagne, all those things. Yeah, I think honestly, buyers seem pretty comfortable with this. It really comes down to how we approach this and how we present it to the industry and to consumers. I've come up with a initiative we could work under, call it the Triple P or the P cubed, the pursuit of the perfect peanut, and even document how we're going about this with the scientists. And I'm not talking about documenting from day one, but once we start having some successes with this, you could put it on a podcast. I mean, I know Scott Monfort does a podcast update, how that's going, or you could do your own, YouTube update on a monthly or quarterly or whatever basis, and try to get that information out there, have the complete transparency, try to educate people as you go along this process. Here's what we're doing for you. Here's what you're getting. And I think that would go a long ways, no matter, whether you're trying to increase yields, increase disease resistance, or improve the flavor or the traits of the peanuts. If you document it along the way and really try to be transparent, I think it makes it an easier sell once that science is completed.
Thank you, John. Josh, you shared several, climate change and other things that you think the gene editing can assist with. What traits would be the most value if they could be improved on at the seed level? And just a follow-up question to that, I guess. What would a manufacturer need to see or validate before accepting a new variety that maybe meet those needs?
Okay, so yeah, just like you said, Chris, some of the traits that we see as being most valuable are around climate resilience and aflatoxin resistance, so around food safety. You can kind of bucket them maybe in two buckets. You have things that happen on the field, which I just mentioned, and then you have the end consumer. So things that would be important for the end consumer would be around flavor. We could do a lot of work, spend a lot of money, but ultimately, if it's not value added to the consumer in terms of stable shelf life, flavor, consistency, then we run some risks there. As far as what we would need to validate, we'd want to see multi-year consistency, multi-region performance. We want to see the value in the supply chain, and then on the benchtop level and consumer level, we want to see flavor, consistencies, processing performance, and not to mention food safety as very important to be able to validate.
Great, thank you. Josh, just sticking with you to change up the order here a little bit. What is your biggest concern regarding the use of gene editing in peanuts?
So I think Wayne did a good job in his presentation about calling out some of the concerns. Regulations are one of the biggest ones. There's, it's a little foggy there in certain areas of the world. We IP ownership is probably a big one. We can probably go back in history and see where, the IP discussions maybe didn't happen soon enough, and it's kind of tripped up some areas. Ideally, if we had strong alignment on how IP would work, it wouldn't stifle innovation. It would only enhance it, as opposed to say one sector owning everything. And then consumer acceptance and consumer perception. That's another big one for us. We want to make sure that the consumer likes it and they're going to continue to buy it, and there's not a lot of challenges at that level.
Reagan?
So I think one of the biggest challenges is, like Josh was saying, getting that consistent buy-in from consumers and manufacturers, especially if we're producing products where gene editing has to be regulated separately from conventional products. So segregation is obviously a really big cost for shellers. Whenever you have to segregate any product that's different from another type of product, that incurs a lot of additional expenses in storage and handling and transportation, shutting down the shelling plant, bringing it back up, making sure that everything's cleaned out. And so we have to really consider not just gene edited versus non-gene edited, but also if we're getting into some of these other characteristics, like John mentioned, like a really high protein peanut or a peanut with superior flavor, all of those introduce different segregation categories too. And so all of those things kind of have to be considered whenever we're looking at what types of traits we want to look at.
Just as a follow-up on that, Reagan, what would make it, easier to manage that supply chain? A unified voice on what traits, or what, so please.
Yeah, for sure. If we can all get aligned on what those characteristics are, right now we just segregate for the oleic acid trait in peanuts, so high oleic peanuts versus conventional peanuts, and just having those two different segregation categories can be very difficult. So for sure we need that alignment, but also having a gene edited product it may or may not be where we go with our research priorities. So if we decide to prioritize research and gene editing that doesn't necessarily lead to a gene edited product, then that could also make it easier because we wouldn't have to segregate that separately.
And same question, but also from a grower perspective, what would need to be proven before growers would plant it, essentially? So biggest concern, and then what would need to be proven for the growers before planting it?
I feel like consistency would be number one. We could not have yield drag or any of the other problems that have been, when high oleic was first introduced, we would not be able to suffer. We would not want to suffer through that. I think one of the interesting questions is, like Josh put out, was what are the intellectual property? How much is it going to cost us to plant this seed? We found with corn seed, cotton seed, soybeans, it gets very expensive to buy a bag of seed because of the intellectual property costs due to the manufacturers realizing that we are a different industry and that we do things differently than, say, a Bayer or a Syngenta or someone of that nature. But what is it going to cost us, and what is going to be our return? That would be one of the first questions I would like to ask.
Now we're going to jump around on this one. So we're going to start with John. Even if the science works, adoption is not automatic. We kind of heard that with regulatory aspects or complexity to supply chains, consumer acceptance, things of that nature. what would you need to see before gene edited peanuts could be moved from a research to practical consideration? And just specifically, what should we avoid doing as an industry to maintain trust throughout this process?
It goes back to what I was saying before. I think transparency and education along the way. This is not something that you just kind of throw in people's laps once you've done all the research and hope they accept it. Like I said, I think if you educate along the way and document your progress and have people somewhat invested in it, then it makes that adoption much, much easier when you reach whatever the ultimate goal is we set and what we hope to get out of this.
Reagan? What should
Yeah,
we do
so
before? Oh, go ahead. Sorry.
So, yeah, I was just going to say, I think that regulatory clarity is really what's most important. It's just understanding, what's this research resulting in. I think that could be handled in different ways. If we have a product that does need to be processed or labeled differently, and that's handled a different way, if we do or do not have to do those things. But consumer acceptance is definitely a priority, and we would have to have a pretty beneficial trait to get any type of market acceptance. And so I think we could really benefit from doing some consumer research on what consumers actually accept, because even if we have unlabeled products, those could be scrutinized by the public. I think we've mentioned that a couple of times. And so we just need to make sure that we have all of our bases covered before investing our research dollars, and making sure that those are used efficiently.
What would your segment need to see before gene edited peanuts could be moved from research to practical consideration? And what should we do? What should we avoid doing as an industry if we must maintain trust? Dan.
I like everyone's answer so far. I do think the transparency is perfect, but I think Reagan's answer, consumer, checking with your consumers, but I think consumer education is a part of that also. I know the National Peanut Board has done a pretty good job of their programs of promoting peanuts, but also educating consumers about where the peanuts come from. I think you'll have to utilize more aspects of those units that are already in place.
Thank you, Dan. Josh.
Can you repeat that question again, please?
Yes, absolutely. What would you need to see before gene edited peanuts can move from research to practical consideration? Specifically, what should we avoid doing as an industry if we want to maintain trust?
It's very similar to, I think, what everybody else has said here about regulations and consumer acceptance. I think to add to that, early stakeholder involvement, as well as like farm to manufacturing alignment. How are we going to do this all together? And things of that nature.
Well, we'll stick with you on this one then, Josh, as the next question. So a trait cannot help one segment while creating a problem for another one, right? Which you're essentially speaking to. How do we make sure a future peanut innovation are valuable across the full chain? And then specifically, what role should the Peanut Research Foundation play within that?
So come together with one voice, be transparent. And like I mentioned earlier, we want to have a forum where we can all discuss at different levels of the supply chain together what our science-based beliefs are and how that's going to benefit the peanut industry as a whole. We want to have shared trait priorities. So what we're working on at the university and breeder level, is shared across all the different industries so that we can all benefit. And then we want to have cross-industrial acceptance as well as alignment on the validation component, how we want to validate this at all the different levels, and when we get our results, we all agree that, it's time to make the next steps.
Thank you. Yeah. A trade can't create one problem for one segment and help another. So how do we make sure that doesn't occur throughout the supply chain? And what role should the Peanut Research Foundation play within that?
I think the Research Foundation is a great tool for getting us together, just like we're doing today, to learn what are our possibilities, to learn what are our pitfalls, and to learn what are our next steps. So I think we have to keep the communication lines open. We do have to build trust with each other, and we just have to keep working at that every day.
John?
I would say just testing the results along the way. I mean, if you think you've increased the protein, but it has a effect on the flavor, we won't know that until we process the peanut and turn it into the product. So along the way, you just need to have the various manufacturers kind of test these things out as the science is going on to make sure we're on the right track. I mean, it's not just trying to get to solving the problem, it's seeing if the solution actually works as we're going. Going back to your previous question, I think one thing that'll help with consumer acceptance, I don't know if it's Dr. Parrott or Dr. Baker that was talking about eradicating blindness and deafness. You know, those kind of things with gene editing will make this more mainstream and help us, even though it's outside of our industry. So I think as people see the benefits of gene editing over the next decade, it will become more generally accepted naturally. So anyway.
Reagan. Supporting the
Yeah.
full supply chain. Go ahead. Yes.
Yeah, so I think alleviating some of these risks of creating problems across, like, in different parts of the supply chain, I think that a lot of that can be alleviated by just communicating between different sectors of the industry, and just understanding what the problems are and how the supply chain works. Kind of like what John was saying, if you know, we need a higher protein peanut, maybe that doesn't mean that there's just one variety of peanut that's higher protein. Maybe it means that we set a threshold for the varieties that we do produce of what protein should be, and that can eliminate the need to be able to segregate separately. And so it's just that communication piece, I think, is going to be important as we develop a lot of these solutions that we're talking about.
Reagan, can you speak to more about how you think the Peanut Research Foundation helps with that communication piece? Is it the vector of that?
Yeah, I think so. And I think just because we have stakeholders that are represented across each of those segments of the industry, and we're all looking into, you know, what do we want to see happen across the peanut industry, I think that those conversations definitely come up, and that we're a really great source to be able to coordinate a lot of that information, and figure out where we can align our priorities.
We got some time that allows for this one because John's talking about blindness and YouTube pages and all these other things. How should the industry talk about gene editing in a transparent but not confusing way in your mind?
Loudly.
Oh, okay. What does that mean? Like, I mean, consistent, more often,
Yeah,
transparently. Say it from the back, right? So.
I mean, as I was saying, with, you know, documenting along the way, just be constant with the flow of information as we get into this. And like I said, I don't think it's going to be from day one, but once you've kind of built up some results, you start releasing them and just keep trickling them and putting that information out there and trying to keep people engaged.
Josh, from your perspective as a manufacturer, loudly, how does that, how do you feel about that? Transparent but not confusing. I say I kind of agree with John, but what does that mean to you when you hear someone says loudly?
I think clear, concise communication often is one way to go. you want to be reliable, you want to be sincere, and you want to be competent. You want to build trust with the consumers. That's probably a really important thing. Being transparent, uh, making sure they know that they see behind the curtain, and can understand what's going on, and all the research and science that's gone behind this, and all the dedicated committees that have helped move this along. And it's just not a knee-jerk reaction, or they see some, some type of flash negativity on the news.
Reagan, we'll start with you on this one. We're 10 years in the future. You get to be aspirational. If gene editing becomes a responsible and accepted tool for the peanut improvement, what is one outcome you hope it delivers for the shellers, and one outcome you hope it delivers for the whole peanut industry overall?
Yeah, so I'm going to kind of expand on that question because I don't know if I'll get to answer again with time. But I kind of foresee if we amp up this gene editing type research in peanuts, that this is going to come kind of in two different phases. And this first phase is really using gene editing more as a tool in research rather than the process of producing gene edited products. And I think that, especially with what we've done with sequencing the peanut genome, and now all of the tools that we have to sequence genomes quicker and cheaper than we have in the past, that just using gene editing for, to help conventional breeders out, I think is this first phase of what's going to happen. And then expanding on that, we can actually, more easily implement gene editing to the point where it might be gene edited products might be integrated into the peanut industry. And if that happens, I would hope for shelling specifically that we can make some quality improvements to some of those post-harvest characteristics that I talked about earlier. And then overall, just be able to allow growers to use less inputs and produce a better crop, and makes us overall more reactive to emerging threats in the market. Kind of what was mentioned earlier about Argentina having, this type of technology, and they might, get to this type of technology and be able to distribute things quicker than U.S. peanuts. I think 10 years down the road, I would hope that the U.S. is competitive with those types of markets.
Thank you, Reagan. Dan, aspirational, 10 years in the future. What would you like to see hope delivers for your segment and delivers for the whole peanut industry?
I hope that 10 years from now, we're still telling our great peanut story, and we do have a great story to tell. But I think that I will be making less trips across the field. I think I would require less inputs. That would be my hope and my goal of this whole investment in time and money, that we would have less inputs. We would be able to have a more consistent crop with quality yields, but also the quality profitability. That's still the key. We don't have to make all the peanuts in the world, but we need to make them profitably.
Thank you, Dan. Josh, 10 years in the future. One for manufacturers, one for the whole industry. What would you like to see?
I think from manufacturing, we'd like to see a peanut crop that's highly resistant to climate change, to drought, heat tolerance. We want to see a consistent quality for the consumers, and we want to see the safety of the crop really enhanced, where those challenges that I just mentioned are really pushed down the totem pole, and we can go about the manufacturing side of the supply chain. And then from the industry, I think in general, if there's a lot of focus on sustainability, you can achieve that a variety of different ways. The more sustainable the crop is, all sections of the supply chain are going to benefit. And we want to see, fewer losses and less risks as a whole industry.
Thank you, Josh. John, you're going to say it loudly. What, 10 years from now, one outcome to help your segment and one for the peanut industry overall.
For the peanut industry overall, I mean, what everyone said, just lower or better disease resistance. Even if we could do away with aflatoxin, that would be huge, or tomato spotted wilt, any one of those. One nobody's really talked about, but I think would be interesting is shorter maturities. If you could have a peanut crop where you could cut maturity, I mean, half would be incredible. But that would give a grower an option to back-to-back 2 row crops during a growing season. Maybe
Or more
help
consistent maturity.
Yeah, or more consistent maturity. And lastly, just a better product. Whether it's more flavorful, has better traits that consumers want. I want to see a peanut that people want again and don't let sit in their pantry in the jar for months and months and months at a time like they are right now.
All right. Well, y'all are very efficient with your answers. So with that, I want to thank you all for participating in the panel. One thing that I think is very clear is that this is not this technology doesn't bring a single project, right, or a single trait. It is really a strategic capability that we're talking about, and only works if we're aligned as an industry. So we will have to stay connected and focused on solving meaningful problems along the way. And hopefully, and it shall be, right, the Peanut Research Foundation is the hubcap that spokes out the wheels that makes this successful. So with that, I thank you all for participating. John Moore is going to send you all a thank you basket. I'm just joking, John. This is recorded. I shouldn't make jokes to John while being recorded, because that means he feels obligated to do it, maybe, or he's going to send me the bill. Either way, I want to thank you all for participating. That does come to the conclusion of our gene editing town hall. So with that, again, next steps is for the Peanut Research Foundation to meet as a board and really discuss this opportunity a little bit more clearly and holistically, and have a few more conversations on that as we move forward. So with that, I want to thank you all for participating. We're ending about 20 minutes early, which is usually pretty great when you have a long meeting like this to catch up on emails, or Dan Ward would maybe do one less spray in the field potentially here. So thank you all for your attention and taking the time to join us today.
Thank you.
Thanks, everyone. It was really great.
Thanks very much.
Bye.